Hello and welcome to SETI Live. I'm your host, Beth Johnson, Communication Specialist here at the SETI Institute. Thank you to our viewers from around the world for joining us today. Please let us know where you are watching from. And also, welcome to listeners on the podcast version of SETI Live, available on most podcast platforms. Now, the project we're here to talk about today is the INVADER project, which researches ways to improve life detection in ocean worlds by using our own oceans as an analog. So they're working on flight-ready instruments, and they're trying to find out how they can do these in situ analyses. And today I am happy to have with us Dr. Bonnie Teece, who is working on the INVADER mission, and in particular is working on these microbial traps that were left on the ocean floor in the Pacific Ocean at Axial Seamount and have been brought back to the surface and analyzed. So Bonnie, welcome in. Thank you for joining me today.
Thank you for having me.
So this is kind of exciting. I really like this. So did we discover new critters? Anything new that you found in this?
Not that we can tell yet, but we did find some signs of life from the microbial trap. So there's stuff there, but we're not sure if it's anything new.
Okay, so let's start at the beginning here. So the INVADER mission, we've talked about this on SETI Live a few times. Pablo Sobron is a part of it. He's here with us at SETI, and we went down. We were using lasers. We were doing in situ analysis. There was all kinds of really neat science going on. So how did this work for your part of it? Did you get to go out to Axial Seamount and pick up these traps? Like, so what was your part? How did you start off here?
Yeah, so I did get to go out to Axial Seamount and pick up these traps, and these traps weren't part of any of the analyses that Pablo was doing on the seafloor. So instead, these traps were left about 2 years ago by the former postdoc in INVADER, Laura Rodriguez, and she placed them in 2 different areas in Axial Seamount. And so these were low temperature areas of hydrothermal venting. So hydrothermal fluids, they can get up to 400 degrees Celsius, but that's too hot for life. Nothing can survive at those temperatures. But there are some areas that have less hot fluids, so about 23 degrees Celsius or even lower. And so that's more attractive for life. And so she placed some microbial traps in 2 different locations, and we left them there for 2 years so that they could accumulate some microbial yummy goodness. And then 2 years later, I got to go on the cruise and go collect them and bring them back to the laboratory so we can analyze them and see what grew on them.
Okay. So you got to go out there. That's really neat. I got to do the same. So I'm like super excited to talk to someone else who knows what it's like to like be on a research ship and go entertain yourself at a volcano. Why are hydrothermal vents important in this circumstance?
So the cool thing about hydrothermal vents is that they provide an energy source that isn't the sun. And so when we get to the outer solar system, we're not in a really good place for photosynthesis to occur. We're very far from the sun. And when we look at these icy bodies that are targets like Enceladus or Europa, they've got these big, huge ice sheets, right? And so there's no sunlight getting into the ocean. So hydrothermal vents provide an energy source distinct from sunlight that could help fuel life. Now, we know from our own planet that when you look in the early Earth's record, 3.5 billion years ago, and you look at fossilized life, we often find it associated with hydrothermal activity. And so we know from ground truthing, our own geological record to where we look for life elsewhere, that these hydrothermal environments are highly conducive to early life and to life being able to get some yummy goodness.
Okay. So I love this because, you know, one of the things that we here at SETI are very, you know, fond of trying to understand is the origin of life on Earth and elsewhere in the solar system and the universe. So you're Australian. You probably know about like the microbial mats that have been found in Australia. How does this relate to those kinds of discoveries as well?
Yeah, so when I was in Australia, I was actually working on these early fossils from Earth's history. So I worked at a 3.5 billion years ago in the Dresser Formation, which are some of the best preserved, most generally accepted evidence for early life on Earth. So in the Dresser Formation, we have these beautiful preservations of layers of microbial mats that we call stromatolites. And these are all found in hydrothermal environments. And in fact, the Dresser Formation records an environment that doesn't just have deep ocean hydrothermal vents, it also has land-based hot springs like Yellowstone as well. And so we have this like mixed inputs of different kinds of hydrothermal environments. So we know that when we look in this record of early life, we're finding a lot of things near hydrothermal environments. And in fact, one of my favorite places in these ancient rocks is this curl of barite, which is a rock that's precipitated from hydrothermal activity, and you have stromatolites growing directly underneath it. And we have this preservation of just that one-to-one association.
Oh my gosh, my geologist heart just like stuttered. I'm like, I want to see this. So, all right, you went. We're looking basically for ways to detect microbial life, ancient life. We're doing this here on Earth as sort of an analog for these icy ocean worlds that may not have sunlight. We're talking Europa, Enceladus, Ganymede. There's a whole bunch of subsurface oceans we've discovered. So you went out, you got these microbial traps. This is the first time really you've gotten to talk about this outside of what you've been working on. So what have you found? What are your initial results?
Okay, so let me explain the microbial traps and why they're super relevant to these ocean worlds. So basically what they were these 4 PVC pipes that were cable tied together. And inside them, there was also like a mesh to keep organisms that were too big out. So no eukaryotes could get inside them or big mollusks or anything like that. And so it was only allowing organisms of a certain size into the trap. And in each of the PVC pipes, there was a different substrate. And so we wanted to see what kinds of different substrates the microbes were attracted to. And so they were each relevant to astrobiology in some way. So one of the traps had basalt in it, and basalt is actually the bedrock of Axial Seamount. It's a common rock that we find all over Earth and Mars, and we think it's very present on the seafloor of Europa, if Europa has an ocean. We also had pyrite, which is an iron sulfur mineral that's also found very commonly in hydrothermal vents and is a mineral that preserves the earliest signs of life as well on Earth. We had a chondrite simulant. So a chondrite is a type of meteorite. And actually, they think that the core of Enceladus might be made of chondrite. And so seeing what microbes like the chondrite tells us about, hopefully, about Enceladus. And then the fourth mineral or rock type that we had was something called serpentinite. And serpentinite is an alteration mineral, and it's formed when olivine undergoes transformation, and it produces heat and hydrogen and turns into serpentine. And so this is a hugely important mineral when we think about life detection elsewhere or hydrothermal vents, because the heat that goes off from this transformation process can actually power hydrothermal fields. And so each of these different substrates we collected is important for different reasons and can tell us a bit more about ocean worlds in different ways. So when I brought the samples back, the first thing that I did was to see which samples had the most organic carbon. So one of the great things about this study is that the people who were working on it earlier made sure that we had controls. So we have the same minerals, but that they didn't go down to the seafloor. And so we can measure what we find in those minerals that didn't go to the seafloor and the ones that did, and see what the difference is. And what we found was almost all of the minerals and substrates had a huge increase in organic carbon, except for pyrite. The microbes did not want pyrite at either location that we had the microbial traps at, which is very surprising to me. But what was most surprising to me was that serpentinite was by far and large the most attractive mineral for microbes at both locations. And so when we looked at the results from the serpentinite, we could see that it went from having like a very low TOC that was, equivalent to something you might find from the ancient earth, to being over like 1.5% TOC. So the bugs were just loving what was going on in the serpentinite. And so then I ended up looking at the serpentinites with Raman to see what we could find. And we could actually see fresh biofilms colonizing this serpentinite, which is just absolutely incredible, right?
So explain for the audience what TOC and Raman are, so that we're not losing them in the terminology there.
Thank you. That's a great point. So TOC is total organic carbon content. So we can have inorganic carbon, or you can have organic carbon. And the reason that organic carbon is important is because all life that we know of consists on organic carbon. Mm-hmm. Not all organic carbon is biological, but all biology that we know of uses organic carbon. So it can be an indicator that there are some organisms growing on something or present in something. Mm-hmm. Raman spectroscopy is one of the techniques that we use, and this involves looking at light and its interaction with a molecule. And the reason that Raman is super cool for lots of reasons. We use it in lots of different industries. It's used in the cosmetics industry for quick characterization of things. It's used in the ancient earth. It's used on Mars. So Raman is currently right now actively searching on Mars as part of the Perseverance rover and looking for organic molecules and minerals. And what's exciting about Raman to me is that it tells you information about both the mineral and the organic content. And so we can start to understand more about the picture of the organics and the minerals together, which is very cool.
Excellent. Okay, I'm going to take a second to welcome some people in, and we're going to get back to our topic. So let me welcome people in from San Francisco, not very far, Texas, Germany, Guten Tag. It's probably evening. So hello, Julia. We have, elsewhere in Germany, North Carolina, Pennsylvania, Kansas, Norway, let's see, the UK, Louisiana, Chile. So a nice global sample here today. I'm going to answer one quick question. Helen, Raman is spelled Raman. So when you're looking at Raman spectroscopy, that's what you're looking at. I know. Ramen, noodles. Okay.
I know, it always makes me so hungry when I'm trying to work on it. I'm just like craving noodles all the time.
There are some things that just trigger that. So once again, I am talking to Dr. Bonnie Teece, who is working on a section of the INVADER mission looking at microbial traps that were left on the ocean floor of the Pacific Ocean near a fantastic place called Axial Seamount. So what can you tell us about Axial Seamount and why this location was chosen?
So Axial Seamount is one of the most well-studied hydrothermal locations in the world. So even though we know that there are lots of hydrothermal locations in the world, many of them still haven't even been visited or confirmed. We just know that they probably exist based on a lot of other scientific data. And then there are many sites where people have been to a little bit, but not a lot. But Axial Seamount is one of 3 places in the world that has what's called a cabled array. And so that means it's continuously monitored. And so we know a lot about what's happening in Axial, which is not what happens for most of the seafloor, right? We have not explored most of Earth's seafloor. But when things change at Axial, we know because of the monitoring that's happening. And this is one thing that's super important when it comes to having an analog site for other planets or moons, is that you need to understand your analog site and its limitations very well, because nothing is going to be a one-to-one comparison for somewhere else. And so, for instance, one of the things that we know about Axial from decades of research that's being done there and decades of monitoring is that after eruptions, the fluid chemistry changes. And so if you have changing fluid chemistry or changing conditions, that's going to affect the samples that you're baking down there, or in our case, that we were baking down there. And so by picking this extremely well-characterized site, that becomes a really firm context by which to start to try to apply to places that we don't know as well.
And so far, what do you think you can apply to places like Europa and Enceladus that you have learned from these two microbial traps at this point?
Well, I think that the findings of serpentine are really exciting for thinking about Enceladus, for instance, because Enceladus, its chondritic core, it likely contains olivine. And so that olivine is likely being altered to form serpentine. And they think that's what's fueling hydrothermalism at Enceladus, if it does exist. And so knowing that serpentinite is an attractive beacon to find life in these hydrothermal events with instruments that we can send on a spacecraft, that becomes a really good place, I think, to start looking for signs of life. And it shows that it's a place where microbes seem to be pretty happy. They seem to get enough to eat.
Being a Californian, serpentinite, of course, very important here in California. We have a ton of it. I have hiked through so much serpentinite, so I'm really excited to see that it has even further implications in the search for life. So I really like that news. That's exciting to me. So we are getting some audience questions in, so feel free to ask those in the comments, and I'm taking a look at them. So let's start, and let's see. Oh, this is a fun question, just because we are all about these here at the SETI Institute for random silly reasons. But Patricia is asking, do tardigrades live in the vents you describe?
That's a great question. I have never actually thought to think about whether tardigrades do live in these vents. I always, and this might be a problem of myself, is that I don't think about tardigrades unless I'm thinking about space, because I've always only learned about them in the context of their extreme ability to survive, and that they can even, desiccate in space. I've never thought of them in a deep ocean vent community, but their nicknames are water bears.
I would not be surprised. I don't know the answer either. We're going to have to go look that up. So thank you. You've given us a thing to go check out there. All right. Let's see what else. Ricardo is asking, given the different types of hydrothermal systems, deep sea vents, shallow sea, intertidal, hot springs, how can they be? Oh, sorry, that changed. How can they be correlated to primitive Earth or extraterrestrial bodies?
Yeah, that's a great question. And the answer is it depends on which one you're looking at, because even when you're looking at different deep ocean hydrothermal events, whether they're alkaline or whether they're acidic, might change what they're applicable to, and the same with their temperature, and the same with the minerals that make them up. So, for instance, when I did my PhD, I actually worked on land-based hot springs and their application to places on Mars, like home plate at Columbia Hills. And that's because they're silica rich, they're in these high UV conditions with lots of irradiation. But you wouldn't compare necessarily a deep ocean hydrothermal environment to that. So it really depends on specifically where in the planetary body you're trying to target and what you're trying to understand. So one of the things about Europa, for instance, is there's an extreme amount of pressure in that ocean, way more than our ocean pressure. And so we have to understand that when we're looking at an analog for Europa, that's a limitation of our analog. It's not, unless we're doing a laboratory study where we can simulate that pressure, we're not getting a complete one-to-one. And so I think we always have to think exactly what is the thing that we are trying to learn about, and how does that relate to the planetary body that we're trying to study, and what are those limitations? And we really have to set out those limitations clearly, because there's always going to be limitations when we're thinking about this. Even when we think about the primitive Earth, obviously the modern Earth is much more oxygenated than the primitive Earth was, and we have much more active plate tectonics. We have continental mass now that we didn't have in the primitive Earth. And so in some ways the primitive Earth is an alien world of its own.
Oh, I like that. That is going to be one of my pull quotes from this. The primitive Earth is an alien world in its own. That is a very solid point to make. I think this other question kind of goes along with this, and I think instead of comparing the worlds to primitive Earth, it looks like Nazaire is trying to compare them to each other. So how do you compare hydrothermal vents on Europa and Enceladus? And obviously, we're going to need to wait for some more data from Europa Clipper, but how do you compare the types of systems you'll find on these two icy moons?
Yeah, and huge shout out for Europa Clipper. I think we're all waiting to get this data back from Europa Clipper, right? What a cool mission. It's so exciting. So I think it is hard with Europa because so little is known, but they think that we've got this basaltic bedrock and that maybe the ocean's a bit more acidic, and you would be more likely to have these high temperature black smoke events. Whereas with Enceladus, you would be looking at more lower temperature, but still, 100 degrees or so, Celsius, that is, not Fahrenheit, and with a completely different mineralogy, and then also an alkaline ocean. And so on Earth, we know that things adapt very differently to these kinds of environments. So one example I can draw, which is not relevant to Europa and Enceladus, but if we think about the difference between an alkaline fluid and an acidic fluid, one thing we know is that photosynthetic organisms can operate at higher temperatures in an alkaline fluid rather than an acidic fluid. There's a difference of about 10 degrees Celsius. And so even that limitation of how life can survive or change metabolically within these different kinds of fluids is very interesting. Obviously, there's no photosynthesis in an European or an Enceladine ocean, but it's interesting to think about how would metabolisms be affected by those different conditions? What kinds of things would you find at each one? And, the results that we found from our microbial traps indicated that there was microbial growth on the basalts that we collected, similar to what you might find in Europa, but it just wasn't as much as the serpentinites. And so I think that there's a lot of parallels to be drawn from both environments, as they're both icy ocean moons, but then there's going to be a lot of differences as well, which are going to teach us about limits of life at two different extremes.
That actually kind of segues nicely into another question. This is from Arwen. As a researcher, what is your dream analog on site on Earth that you would love to study, and what would it be an analog to? So, do you have one that would be a great analog that you know of for either of these moons? And if not, is there someplace else you want to study that you haven't yet?
So Lost City would be fantastic. The Lost City hydrothermal field is powered by serpentine alteration, and it's a wonderful analog to what's going on in Enceladus. It's also just such a cool place. If you go online and watch the footage from remotely operated vehicles exploring Lost City, it is just absolutely mind-boggling and unparalleled.
And where is that hydrothermal vent located?
That's a good question, actually. I have never thought about where it is geographically. I always just think Lost City Ventfield. I'm not sure.
All right.
Yes. So that would be a place that I would love to study. And so obviously it would be an analog to Enceladus. And then also I have to just give a shout out to the ancient Earth sites in Australia. I've already studied there in the Pilbara, but that's my favorite place on this Earth. And it's an analog to so many places. And it's just a beautiful place to do research.
Fabulous. I'm going to hope that happens for you. So if anybody out there is funding a mission to that, keep Bonnie in mind. Adil is asking, and I like this. So two-part question. How was the equipment placed on the seafloor? And should we modify Drake's equation to factor in the probability of finding life on exoplanets?
So the first part of the question, how was the equipment placed on the seafloor? It was loaded onto a remotely operated vehicle. And so they're called ROVs. And so you load the thing that you want to deposit onto the ROV, and the ROV gets lowered off the side of the ship, and then it sinks down to the seafloor, which takes a long time, like an hour or longer, depending on how deep you're going. We're looking at 1.5 kilometers below the surface of the ocean for our specific sites. And it sinks down, and then it's controlled by a crew. So in the case of the remotely operated vehicle that was used on the cruise I was on, it was the Jason 2. And we got in these giant demountables, and we worked with the Jason crew, who are absolutely fantastic. and they control the Jason. And it's sort of like watching somebody control a rover on another planet, right? Because you're remotely controlling this thing. And so Jason gets down on the seafloor, and then they drive Jason around, and he's got a big claw, and he's got like all of these different things attached to Jason, because of course when Jason goes down for the dive, it's not only to collect what we want or put down what we want, it's to do a bunch of other experiments. It can go down there for like 24 hours or even longer sometimes. So try to pack as much into one dive as possible. But so Jason gets the claw and then reaches in to its belly, I guess, and pulls out the microbial trap and then places it. And so it's really cool. The amount of things they can manipulate with this claw is just mind-boggling. We watched them untie like ropes that had gotten tied up with the claw. There was a crab that wanted to hang out with us on Jason, and it didn't want to get off. It was there for hours, and so they got the claw to try to scare the crab off, and then the crab got its claw, and it was like big claw versus small claw. It's just highlight of my life, absolute highlight of my life. I have a video of the crab like reared up backwards, like trying to fight the Jason claw, which is like so big compared to it.
Oh my gosh, you need to send that to me, because we need to share that. That sounds awesome. I don't think on my trip we saw any undersea battles with our ROV. We had the dock crickets, which is out of Mbari, and so and it is amazing to watch these technicians control the ROV and all of the instruments on it and how they can manipulate things. I mean, watching them pick up like tubes and screws, like we were trying to put something down and they're trying to move this bolt, and it was just amazing. So Adil would like to correct the latter question, to change exoplanets to rogue planets. So I think it's a little outside the scope of this. So don't feel like you have to answer that. But I will say that yes, I think we should add the existence of rogue planets into the Drake equation, but I think that kind of comes under how frequent exoplanets occur. And I think that rogue exoplanets will occur with about the same frequency. So, it's a similar thing. But yes, it should be accounted for. I will stand by that. So I want to just welcome in a couple more people. So let's see. What did I see? I saw North Carolina, Poland. I think that's it. But I was so excited to see this. And they would like to let you know, it looks like the Lost City Hydrothermal field
it's Atlantic Ocean. Atlantic Ocean.
Yeah.
Yes. I remembered while I was answering the question, and I was waiting to let you know, because it's like when you're trying to remember someone's name and then later you're in the shower and it just like pops up. It's the Atlantis Massif at the Atlantic Ocean. Thank you everybody for bearing with me during that.
I like this. The old Lecomte High School and Museum is asking not a mile and a half, kilometer and a half down, is it completely dark there? And the answer is yes.
Yes, except the Jason lights everything up. And I think this might be why the crab was so angry. Because imagine, it's like when you wake up in the morning, right? And someone turns the light on before you've fully woken up and you're like, oh. I think that's what we did to the crab and all the other life forms down there. And so they were just like, what is this brightness? We've adapted to darkness and crushing pressure. What have you done to me?
We've adapted to darkness and crushing pressure like every grad student ever. Oh, I mean, sorry.
I related to the crab, yes.
Yeah, it was a meme that was posted on several doors in our grad department for geology. We were just like, no, we all feel this. That actually kind of leads into another question Arwen had. So what does live in the vents? What sort of life did you see around these? I mean, you're talking microbial. So is there anything you can specifically speak to that live there?
Yes, actually. So we had to have the mesh on our traps to keep everything else that lived there out, because there's so much stuff living. And so stuff lives on the vents, but not in the vents because it's too hot, right? The temperatures in these black smoker fields are too hot. We were on a mount, which was a diffuse mount. And so this was percolating the 23 degrees Celsius fluid through a mound of sediment rather than a through a chimney itself. But in the mounds and on the chimneys, you have like a wide variety of limpets, which are a type of mollusk. And actually, when we brought the traps up, I felt like the limpet destroyer because they were covered in limpets. And I was trying to like pick them off and like not with my hands, obviously, but with like tweezers and put them back into like the box and like let them back into the ocean. But I knew it was too late for them. So I'm sorry, limpets. I think that we may be at war now, but I love you very much. There was also a bunch of tube worms, which are really cool, and lots of other like plants and things like that. And then you had like the macrofauna. So I talked about the crab, but we had jellyfish and starfish, and there was also squids. And so if you had like the shift in the middle of the night, everything was like super active down in the seafloor. And so you would just see like squids coming along and like crews and everything. It was very cool. Actually, seeing the sea life was the part of the most exciting stuff that we were seeing. But the chimneys are teeming with life, like absolutely teeming with life. Anywhere that life can be down there, it's down there, because this is the main source of energy for these small things.
Yeah, I have to say that was one of the kind of cooler things. I think that's why the biologist on my mission really like just disturbed me, because he would just grab things and take them up to the surface, and I'm like, oh, but it's been down at like 1,500 meters and 2,000 meters, and it didn't probably survive that trip. And I really don't like killing the creatures, so I understand. Like, I feel so bad. I could not be a biologist. Never in a million years could I be a biologist. Rocks, any day of the week. I will pick up rocks.
We take them from their homes. What if they miss their home?
I know, exactly. I mean, microbes are one thing, but like bigger life, just it really starts to get to me.
Yeah.
So Bonnie, we've made it through our half an hour. So now I'm going to ask you one final question. What's next for Bonnie?
Oh, well, I'm going to finish analyzing these very cool samples that we've got, and hopefully come up with some cool results to present to everybody in a scientific paper. But we're also growing chimneys in the laboratory. So that's really fun. We've been growing different chimneys with mineralogy that are similar to these black smoker chimneys, and we're growing them in the absence of life. Which you don't find at the seafloor, right? And so really exploring the transition between these two things, between where there's something teeming with life and where there's something not teeming with life. And for me, I'm just really interested in like what happens to a life form when you look for signs of life and it's been like really destructed and it's really hard to piece together. So if you have this 400-degree hydrothermal fluid traveling over like preserved cell membranes or something like that, how do you recognize it? Can we recognize it? And what does that mean? And so that's like the main question that drives me. So we'll see where that takes me, but that's what I'll be thinking about.
I love how your work is such a blend of different areas of geology. You've got some microbiology in there, but you also have, paleontology and sedimentology, and how, trying to figure out how all these things interact together. It is a lot to know.
Well, actually, a lot of the time I'm like, should I call myself a geologist? I don't know. If somebody asks me about mineral identification, I can't do it. But I can't call myself a chemist either, because I don't understand reactive chemistry. So it results in a lot of like existential crises.
The term planetary scientist like covers a multitude of sins, I think. So I really... I kind of embrace it, because I'm like, I mean, my undergrad's in physics, but it was really astrophysics, and my master's work was in geology, but it was, but I do a lot of planetary. So yeah, no, I feel that. I absolutely feel that. So, well, Bonnie, thank you for joining us today. This has been fascinating. I'm very, very excited for you on several levels, and I'm excited to see more from the INVADER mission work. Once again, to remind everybody, and thank you for watching today, the SETI Institute is a 501c3 nonprofit. So your subs, your bits, your super chats, your stars, they all help. Once again, Julia, thank you for the stars. Very, very grateful. If you are interested in supporting us in a larger fashion, you can go to SETI.org, and there's a big red Give Now button. You can click on that. And we will be back next week. Let me just double check what we are doing next week. We will be back next week to talk about, I believe, an exoplanet, night side clouds on WASP 43b. So one of those amazing WASP planets that I'm always fascinated by. So come join us next week for that conversation. And again, thank you everybody for joining us. And thank you, Bonnie, once more for being here today. Much appreciated.
Thank you very much.